Okay, so in this video, we're going to look at how we can apply CRISPR DNA technology. So in terms of the study design, the last video looked at the actual function of CRISPR-Cas9 in bacteria, and a bit of background into that. If you haven't watched this video, you need to definitely watch that first. This video goes into how we can apply this in editing an organism's genome. So you'll know that you're successful in this lesson if you can describe how CRISPR-Cas9 technology can be used to edit another organism's genome, but you also need to be able to outline or identify examples where CRISPR-Cas9 editing could be beneficial. So this is a screenshot from, again, your textbook. The purple part was used in the last video when we were looking at CRISPR-Cas9 and how that the Cas9 enzyme works to cut specific sequences of DNA. So what scientists have been able to do is to artificially, create these Cas9 enzymes with guide RNA to target any DNA sequence that we want. So we might pick a sequence and say, all right, let's try and cut some DNA there. We can put it into the machines, make a specific sequence of DNA and insert that into the Cas9 protein. We're then able to potentially add into that same little test tube a new gene that we've sequenced again artificially with those complementary ends. So as the cell then of whatever organism we're looking at, so you can see here it's been used and the research has been quite widely in mice. It goes from being done in little stem cells in petri dishes to mice before it goes then into other organisms such as humans. So we add this Cas9 with our new artificial targeted RNA sequences. We also add the gene that we want to insert into that organism. And what happens is when that cell then tries to repair that DNA, it repairs it and it inserts that sequence of DNA exactly where we want it. So later on, we're going to learn a little bit about transgenic organisms and how we've edited edited genomes of organisms before. But this is really specific. We can, insert a gene exactly in the genome where we want it to be based on that new artificial guide RNA that we've decided its sequence to cut. So think back to what you already know about cutting DNA and complementary and sticky ends and all that sort of stuff. We can make those sticky ends. We can add our donor DNA. We can, our gene that we want to insert. It has those complementary sticky ends. It's already going to be attracted to that part, and then the cell will repair it, inserting that gene in that specific spot. Very, very cool. And you can see that happening here. That gene just happens to be attracted to that little bit of DNA that's being cut. And then what happens is the cell's DNA, the cell's machinery comes along and repairs it and actually fills in any gaps. So obviously the other side of that, that gene, as well as the bits that I guess were overhanging from those sticky ends. So I'll let you watch it one more time. Feel free to go back and repeat, but it's a really great representation of what is actually happening in the cell. Obviously we can't see all the proteins and stuff that are involved, but you can see that gene that we want to insert into the genome once it's already been cut, being attracted to those ends, and then the cell's machinery comes. So let's look at how this might be useful for scientists today. So if we're looking firstly at genetically modified foods, so some foods are already CRISPR edited. So things like celiac friendly wheat, so they've been programmed not to make the protein in wheat that causes that immune response in celiac patients. Potatoes that don't produce acrylamides when they're fried, high fiber white bread, all of those things are created from genetically modified foods that have been specifically edited using the CRISPR technology. We're using it to study diseases in new ways. So you might be familiar with the BRCA1 gene. It's a gene that's responsible for lots and lots of breast and ovarian cancers in our population. Basically what they can do is they can insert different mutations into that BRCA gene and look at how that responds to how those cancers respond, but also how they might respond to particular types of treatment as well. So, women that come with BRCA mutations, there's the specific type of mutation they have might be responsive to different medications compared to somebody with a different BRCA mutation. And there's also been recent advancements now that use CAS9 variants that don't necessarily cut DNA, but they might turn genes on and off by methylating or demethylating a gene. The possibilities are really endless. So scientists have been using lots of different technologies to modify the genomes of organisms for quite a while, but this is the first that's been really quite specific to certain sequences of DNA, and it's allowed scientists to target genes with more accuracy than they ever have been able to do before. So the possibilities really are endless, and it's a really exciting time to be a geneticist at the moment.